The human tissue lab is very important for understanding of human diabetes because we need to study human tissues to understand what's going on. There are big differences between mouse and rat and humans.
The Human Tissue Laboratory in Uppsala is a collaboration between the Joint Strategic Research Initiative, EXODIAB, and the Nordic Network for Clinical Islet Transplantation. The Human Tissue Lab serves as a contact between transplantation surgeons, the islet facility, and the user scientists.
We have a unique environment, I think, to provide world-class research based on the unique tissues we have, and the unique competence we have, and the unique cooperation that we have.
Receiving human donor material is a great opportunity and a great responsibility.
The importance of donated human material can't be underestimated. It's very important to have human tissue to draw conclusions that are connected to the true scenario in humans. The main task is to transplant islets to a patient that's in need of islet transplantation. But if there is no patient available, and then if there is a consent for research, the islets will be distributed to the researchers that can perform experiments on these islets.
The Human Tissue Lab provides the researchers at the universities of Lund and Uppsala with their most important material, insulin-producing cells. The main tissue handled so far is human pancreatic islets.
When I get the islets delivered by jetpack, they come via airplane and then arrive here in a styrofoam box. Then I distribute the islets to the different researchers, and then they start doing their experiments. Human diabetic islets are extremely important because they are the real thing. It's the mechanism taking place in these cells that we need to discover to be able to find the causes of diabetes.
The Human Tissue Lab characterizes tissues functionally and does global analysis of gene expression. DNA and RNA are extracted from every tissue and stored for future use in the human tissue database.
The Human Tissue Lab is unique from the point of view that each and every time we receive human tissue, we do the same experiment over and over again. The good thing about this is that then later we can go back and we can see in the individual with very well-functioning tissues what sort of genetic variants do they have compared to another individual that has tissue that did not work so well.
Since 2007, the Human Tissue Lab has built up a microarray expression database consisting of 80 donors. This is a unique material for studies of diabetes and has already resulted in numerous high-impact publications.
A breakthrough that we have had in our program is a couple of years ago when we described very strong inflammatory reaction that is triggered when islets are transplanted. To simplify this, you can have a similar reaction triggered when, for example, you cut yourself in the finger. A lot of bacteria enter into the bloodstream, and within seconds the defense mechanism in blood eliminates the bacteria before it hits the heart or the brain, the vital organs of the body. A similar reaction is triggered when we infuse the insulin-producing cells into blood. This was previously unknown, and by discovering this reaction we could also create new means to treat the subjects at the moment of transplantation.
Now the Human Tissue Lab is extending their facility to also include other tissues, for example muscle, liver and blood vessels. To have access to all tissues involved in diabetes from the same person will give an outstanding opportunity to map the genetic and environmental factors.